First and foremost, welcome everybody. We're so excited to have you join us. And just a little bit of context. One, my name's Nicole, but Dennis and I have been traveling this spring and we've had an incredible time meeting community members in person. So we thought it'd be a really good idea to host a virtual session just to get to know more folks and answer any questions that you guys might have. So how this will all work is you can just type any questions that come up into the Q&A box. And first, we'll introduce ourselves. We'll talk a little bit about what we're excited about in drug discovery, and then we'll answer all your questions at the end. So without further ado, I'm going to go ahead and introduce you to the team, and I'm going to pass it off to Dennis who to start.
Great to be here. It's so great to see everybody. Dennis, I guess maybe you can tell us a little bit about the session and.
Yeah, absolutely. So yeah, in this session, we first wanted to make sure that we introduce ourselves, talk a little bit about our roles at Drug Hunter and what we do. And then we'll go into questions, talking a little bit about what we're excited about in drug discovery. So, maybe we can start with kind of a brief round of introduction of, talking a little bit about our backgrounds and what we're currently working on at Drug Hunter. And Dennis, do you want to start with that maybe?
Sure. Okay. Yeah. Just a general introduction. Obviously, I'm Dennis, the founder of Drug Hunter. Lead the team here in trying to serve our community in highlighting the latest in drug discovery. I'm based on the West Coast in the U.S., so it's morning here, and maybe I can pass it on to Shema.
Thank you, Dennis. My name is Shema Kunais. I'm a research analyst at Drug Hunter. I hold a PharmD degree from the University of Jordan, and then I moved to the US in 2018 to complete my PhD in pharmaceutical sciences. And our focus was on the discovery of tool compounds that serve as slack potassium channel inhibitors to treat rare epileptic diseases in infants. Actually now a pharmaceutical company whose name I cannot disclose has licensed that project, and I hope we see something that translates into clinic soon. Now my role as a research analyst at Drug Hunter, I just go through the literature that is pertaining to drug discovery. As our readers know, that we make it in a more digestible and easier to read format. And yeah. That's pretty much it, and I'm going to pass it to Lou.
All right, thanks, Shema. So I'm Lou Pennington. I'm the scientific director here at Drug Hunter. I've been here now for a couple of months. As a scientific director, I'm responsible for leading the scientific content creation that you see on Drug Hunter, which I think you're going to find today during our discussion, if you don't read it regularly, is actually pretty interesting. And so we'll be talking about some of our favorite pieces of scientific content on Drug Hunter. Prior to coming to Drug Hunter, I was a practicing medicinal chemist and drug hunter in industry. I worked at 5 different companies, spanning from small early stage companies all the way to very well-established large pharmaceutical companies. I started at Eli Lilly about 30 years ago. And then I've also worked at Amgen, Array Biopharma, Chimera Therapeutics, in Alkermes. And so I'm really happy to be at Drug Hunter. It's a very exciting time in drug discovery and a very exciting time at Drug Hunter. And so I look forward to talking to you more later today. And I guess I'll pass it off now to Matt Hesse.
Thank you, Lou. I'm Matt Hesse. I'm a senior scientific editor here at Drug Hunter. I've got about 10 years experience in drug discovery before I joined Drug Hunter. And actually, I used to work with Dennis Koester when we were both at Novartis West Coast site in the Bay Area. My experience is mostly in anti-infectives, working in antivirals. I've got a pretty broad spectrum of other experience across protein stabilization, hematological oncology, and also neuroscience. And yeah, my job is to help bring the content that you come to Drug Hunter for. So editing, commissioning, writing articles, and just really looking at kind of what's the exciting stuff that's going on in the drug discovery world that our readers are going to want to see. And with that, I think I'll pass off to Rory.
All right, thanks, Matt. Happy Friday, everyone. My name is Rory, as Matt said. I joined Drug Hunter shortly after the holidays in January. And before I joined Drug Hunter, I was a practicing medicinal chemist at Insight for about 4.5 years. And before that, I obtained my PhD in organic chemistry from the University of Michigan with Corey Stevenson, working on all the blue light chemistry that I'm sure you guys are all familiar with. And so, yeah, my role here at Drug Hunter is scientific editor. And so I also, work very closely with Matt, Shema, Lou on writing, editing, and commissioning and, producing content that you see on our website, which is a lot of fun. And I'll get into more details about that, later in the session. I'm also based out of Philadelphia the Philadelphia area. And so something that's quite unique about our group is we're all kind of based all over the country, which is quite exciting. So happy to join today. We're going to kick it back to you, Dennis Koester.
Yeah, awesome. Yeah, I'll introduce myself too. I'm Dennis Koester. I'm the Director of Industry Research and Relations here at Drug Hunter. So my job is really exciting at Drug Hunter. I get to organize and do events here at Drug Hunter. And really my favorite part of doing this is speaking with other scientists and learning what they're excited about. I also kind of bridge the marketing and the content team. So I help with some content creation, which really is awesome because it keeps me really updated with what's going on in drug discovery. And that's what I'm mostly excited about. As Matt already mentioned, we work together at Novartis. So I've been also practicing medicinal chemist for about 10 years, both at Novartis and at a smaller company here in the San Francisco Bay Area. And yeah, located here in San Francisco, which I really love. Wonderful city, a lot of opportunities here to do exciting research and stay on top of what the biotech world is doing. Yeah, and I think with that, I want to kick it off and ask a few questions of our scientists. And I want to start with Dennis. Dennis, can you tell us a little bit more about why you founded Drug Hunter?
Yeah, absolutely. So, as most of you know, I actually started my career in the biotech industry. My first job was at a startup working on small molecules for cancer immunotherapy. And around in 2018, I don't know if y'all remember this, but the news cycle back then was completely negative for the pharma industry. Every time you opened the news or social media, it was about price gouging and pharma's evil and all this stuff. And I thought this was so unfair because as you all know, there's so much exciting science and incredible innovation going on in the industry, but it doesn't get any attention, right? It doesn't get fair coverage. So I thought, wouldn't it be great if we had a service that was run by scientists for scientists to help the whole industry stay up to date on what's really interesting and exciting? Now, this has grown tremendously. This has turned out to be much more useful in terms of, helping people in industry keep up with what the latest technologies are, the new innovations, the way people are applying new hit finding strategies and photo affinity labeling, all those sorts of things. And it's turned into a really practical service to help people discover drugs faster.
Awesome. Yeah, no, that's definitely I found totally an unmet need. I was using Drug Hunter in my industry career and found it really tremendously useful. So Dennis, can you tell us what's your favorite molecule that was recently covered on Drug Hunter and maybe your favorite article that maybe you learned a lot from? Even you, who basically, in my experience, knows everything.
I don't know everything. I learn everything I know comes from this group at this point. I'm going to cheat and I'm going to pick an easy one first. I'm going to say MK0616, which was our 2023 molecule of the year, obviously. So nobody else gets to pick it. I called dibs. It's just, this amazing story from Merck. Everybody's been working on, this target PCSK9 for decades. As we all know, there's antibodies approved, and there was a lot of criticism about the antibody and oligonucleotide approaches because they're so expensive and have to be injected. So Merck, through mRNA display screening in partnership with Raw Pharma, they found these 1,800 molecular weight starting points to target a protein-protein interaction. And over what I believe is over a decade, they were able to make these triple macrocycles that are orally bioavailable, more potent than the antibodies in some cases, And they recently had phase 2 data that was strikingly positive last year. So to have an oral macrocyclic peptide for such a challenging target is really incredible. As far as my favorite recent Drug Hunter article, I have to think about that one for a little bit. I'm gonna go with your. I think it's your. Correct me if I'm wrong. I think it's your PRMT5 article. I know several people worked on that, and I know we had some great reviews by friends in the industry, Brendan Parr and Eric Newcomb, right?
Yes.
Amazing article. So PRMT5 has been a target that people have been, working on for many years. I remember it was super hot at one point, and then it kind of died, and then now all of a sudden it seems to come back. So it was a really nice primer to catch up on that field and see why this area is so hot. And scientifically, it's fascinating. I think the mechanism of the second generation of PRMT5 molecules, the PRMT5 MTA targeting ones, is super cool. So love that one.
Yeah, no, thanks for sharing that. Yeah, I had a lot of fun working on that too. Talked to a lot of people about this on different events. Brandon, I actually interacted with in one of our dinners. We talked about PRMT5, and then he ended up reviewing the article. So it was super exciting. Yeah, I learned a lot about PRMT5 as well, writing this article, and Matt did an awesome job in editing that as well. Cool. With that, let's go to Lou. Lou, you already talked a little bit about, maybe why you joined Drug Hunter, but can you tell us, what brought you here in a little bit more detail, and maybe what is something that you really love about working here at Drug Hunter?
Yeah. So, I spent a lot of years as a practicing medicinal chemist and a practicing drug hunter, but one other passion of mine during my career was reading the literature and learning about what other people had done to try to do my work more effectively. And so, on top of my day job as a medicinal chemist and a drug discovery scientist, I did a lot of work in nights and weekends writing review articles and perspectives. And I did that out of a desire to learn more, to learn for myself, to do my job better, but also to try to help other people learn how to do their jobs better. Because I got my PhD with Larry Overman, and it was his philosophy that we really need to teach each other. Like, that's part of our job as PhD scientists. It's not just to do the research, but also to teach. And so that sort of teaching aspect has been something that's been pretty important to me in my career. What specifically drew me to Drug Hunter was this very unique opportunity and this very unique niche that Drug Hunter is providing, really, in my opinion, truly transforming the way that we share medicinal chemistry and drug discovery insights. This goes beyond Cortellis. It goes beyond the primary and secondary literature. It goes beyond like the news you can read out there in the world. And really what we're trying to do is not only inform people about the latest advancements in medicinal chemistry and drug discovery, but also really drill into the insights and the transferable knowledge that people can use in their own drug discovery programs to try to help them work more efficiently and more effectively, and try to get those medicines to patients more quickly. So even though, I'm not an active drug hunter at Drug Hunter, I feel like I still have an opportunity to very strongly influence the way that we get medicines to patients.
Well, that's super fascinating. Yeah, I feel the same way, I have to say, about Drug Hunter as well. So can you tell us about your favorite molecule and article recently covered on Drug Hunter, and maybe talk a little bit about what you learned?
Well, so my favorite molecule, this is probably a little bit selfish, but it's one we haven't yet written a Drug Hunter article on. It's actually ALKS 2680, the orexin 2 receptor agonist from alchemy. I won't go on about that. That's my favorite molecule. And I look forward to the day when we can share the chemical structure and the really detailed pharmacology of that molecule. It's really exciting. In terms of my favorite article on Drug Hunter, that's got to be the article we recently wrote on Vividian's Werner helicase inhibitor, VVD214. I mean, this drug discovery story is so incredible on so many levels, from the hit identification method to the lead generation and lead optimization approaches, the chemical structure, the mechanism of action, the tissue specificity, and then the disease indication. So really from top to bottom, this drug discovery story has so many examples of great science and great innovation. And if you want me to go into a little more detail, I'm happy to do that. I mean, cut me off if you will. But one thing that they leveraged for their hit finding approach was chemoproteomics. And I think a lot of people know that chemoproteomics is this relatively new approach to hit finding in drug discovery. And it allows people to look for covalent ligands to proteins. And a lot of times by using this covalent approach, you can actually ligand proteins that were unapproachable using traditional non-covalent types of chemical matter. So utilizing that chemoproteomics hit finding approach was part of this program. They also exploited a very unusual alpha beta unsaturated sulfone warhead. So a lot of covalent inhibitors actually use acrylamides, and those are great. They really are. They really have a nice balance of properties. But for VVD-214, they actually exploited a novel covalent warhead, which was really interesting. And this novel covalent warhead, this alpha beta unsaturated sulfone, is a covalent reversible warhead, which makes it interesting, that binds to an allosteric binding site on Werner helicase. And despite all of that, it's still an orally available molecule, which is pretty incredible. And then on top of that, this compound is not ATP competitive. It's actually ATP cooperative, which lends itself to some further improvements in the pharmacological profile of this drug. And then on top of all of that, given this pharmacological profile, this molecule is actually very specific for microsatellite instability high types of tumor cells, which gives you a great deal of tumor specificity. And this is part of the whole synthetic lethality approach that a lot of people are now starting to re-leverage in oncology programs. So it's really a fascinating program from top to bottom. And if you haven't read this article and you're a subscriber, I highly recommend you grab that cup of coffee and read this article as soon as possible, because you're going to learn an incredible amount.
Awesome. No, I love that. Thanks for sharing that, Luke. No, I actually have to say I also love synthetic lethality targets, and Werner is of course one. And yeah, I learned a lot about, that whole target, and yeah, all of these nice insights that you just shared with us. So thank you. All right, let's talk to Matt a little bit here. Matt, as I mentioned, we worked together at Novartis already, and you mentioned that you had worked in basically a large pharma company and biotech. Can you maybe tell us a little bit about how you got to work at Drug Hunter, and maybe something that you found surprising in your work as an editor at Drug Hunter?
Yeah, I mean, I think a kind of pan-industry experience is experiencing a layoff, right? Which was kind of, so I'm based in Pittsburgh, Pennsylvania, which is not exactly a hotbed of drug discovery, right? And so, I was looking for a new challenge, and Dennis and I used to work together, and we talked. And he said, well, actually, I mean, I think you might want to talk to Dennis, who I know, who I think this company, Drug Hunter, really might be right for you. And I think a lot of people have this experience with Drug Hunters. They recognize it before they really know what Drug Hunter is, right? We have such a striking visual identity, and you kind of see us on LinkedIn, and you're like, oh yeah, I know what this is. And then actually, once you really get to know what Drug Hunter is, it's this whole other world that opens up to you. And so I had a great couple of conversations with Dennis Hu, and this job essentially is, well, your job now is to know everything there is about drug discovery, right? And that's such an appealing thing to me, that it's okay, well, here's this area I've always been interested in, and my job now is to dive in and to kind of learn as much as I can, and then try and distill that down and convey that to our audience, right? And part of that, a really big difference between this job now and when I was kind of a practicing medicinal chemist is, I've worked in some pretty fast-paced drug discovery projects. The deadlines are still measured in quarters, right? It's next quarter or next portfolio review, we need to meet this deadline. Whereas at Drug Hunters, okay, well, this week we need this article out, right? We do a lot of conference coverage, and we want to have, the hot news for our readership and to get kind of the scientific information out as soon as possible. And so I think that different cadence of work has been quite surprising for me.
Cool. No, yeah, definitely. Yeah, that's definitely new and like definitely a very different environment than in both large pharma and biotech. And as for your favorite recent molecule on Drug Hunter and article, what would you say is that, and what have you learned from those articles?
Yeah, I think I'm going to pull a Lou as well, and put a put an article which actually isn't out there, but I'm in the process of writing. This is the kind of Revolution Medicines pan-RAS on, which they recently disclosed at the AARC, which just, it's got such a cool mechanism of action. It's drugging these targets which were just considered completely undruggable, right? I mean, there's been so much work that's gone into G12C, ARAS, but some of these other ones, this kind of molecule 6236 is just, they're inhibiting all RAS, and it's led to some just wild clinical outcomes. And this is one of those articles that's just like, wow, this is so exciting to learn about this and to share this. And I think for kind of articles, I really enjoyed it's not actually an article, but the flash talk we had a couple of months ago from Ollie Phillips on GLP-1 receptor agonists, right? Because I think another thing that Drug Hunter does really well is it really lowers the activation barrier to learning about something. Everyone's heard about Ozempic and these GLP-1, but you're like, oh, okay, I'm interested, but I don't have time to commit to learning that. And just to really get like this overview in an hour, or we've written up as an article 50 minutes over a cup of coffee to really get a good sense of what's going on, and not just a surface level kind of like sense of it. You really go into the science and see the differences between the different players in the competitive landscape. So yeah, I really enjoyed that article.
Cool. Yeah, no, thanks for sharing that. Yeah, I got the opportunity to moderate that flash talk, of course. That was really awesome. And yeah, everybody is talking about GLP-1. Like all of my family members have by this point asked me about GLP-1. So it's really interesting. Typically, people don't talk that much about drug discovery. So it's like really a hot topic that's heavily discussed in society. Cool. With that, let's introduce Rory. Rory, how are you doing today?
Doing really well, Dennis. Happy Friday again. Like I said, happy to be here, and this seems to be a really fun conversation. So thanks.
Cool. Yeah. No, maybe you can tell us a little bit about your day-to-day job. Matt is a scientific editor as well. Maybe can you get the audience a little bit of, like a behind-the-scenes look about what an editor does day to day in their jobs?
Yeah, absolutely. So I'll really just start off by saying every day for me brings new challenges and new learning opportunities, which is really exciting, and it's really something I love about this role. In the morning I could be writing up and editing an article, about NaV1 1.8 inhibitor, and then in the afternoon I could be writing up an article about a CDK2 inhibitor. So just the whole breadth of work that we cover is just really exciting to me, and it just makes this job very exciting and very different every day. So there's always new projects. I'm kicking off and starting, and also just old projects I'm wrapping up and getting ready to push out online. So but at a very high level, my duties fall under 4 categories: writing, editing, commissioning, and producing content, with the main goal of analyzing scientific and commercial developments in drug discovery. And really my main aim is to provide our readers, our audience, with insights and updates on the relevant problems and solutions for their work in drug discovery. And that's really, every article I go into, that's kind of something that we're consciously thinking about. How can we, provide our readers, our audience, with a transferable insight to help solve their medicinal chemistry problems? And so that's, again, a very fascinating aspect of my job and something I really enjoy.
Awesome.
I'm also in a really unique position where I can work cross-functionally with several different departments, and including content. So the folks on this call, Lou, Matt, Shema, as well as design. So we have an amazing designer on our team, and so I always find it very rewarding to work with her in designing aesthetically pleasing posters and very pretty images that you see on our website. So I always really enjoy doing that. I work quite closely with the marketing team to again help to design promotional assets for LinkedIn, our social media platforms, and then I also work with sales a little bit too, and as they need I can jump on sales calls and help out with demos and things like that. So again, this is a very cool opportunity where I'm working very cross-functionally, but at the same time, I'm very much very deep in the science. And, that's something I really wanted in this position. And that's exactly what I get. And then lastly, I have regular interactions with industry scientists for external reviews and facilitating publications of articles from guest authors. And that's, just another really awesome feature of this job where, I can, reach out to really anyone in the field to help leverage their or to leverage their expertise in a certain therapeutic area, indication, modality, whatever it is. And I can pull that knowledge into our articles and really help build my network. And so that's really exciting.
Awesome, awesome. No, yeah, no, those are like, probably people wouldn't have known, that there are so many things that an editor does, which is, I think, why it's awesome that you gave them a behind the scenes look of what the editor does. So as for your favorite molecule and article on drug discovery, can you share a little bit of a nice scientific story with us there?
Yeah, I mean, this is always a very tough question, right? Because, there's always so many, there's, always something that we can choose that, is really appealing. But I would have to say right now, one of my favorite molecules and something that we're very eager here in this year, because we'll probably see, perhaps an FDA approval for this is, Vertex's exquisitely selective NaV1.8 inhibitor VX-548. So this is a non-opioid pain drug that's so urgently needed. And I think we all can relate to how impactful this type of medicine will be and, the impact that it will have for patients. For a long time, we've the industry has been searching for non-opioid pain therapeutic, and this appears to be maybe perhaps the first one that might cross the finish line. What I find scientifically notable about this molecule is it works very rapidly with comparable degree of pain relief to Vicodin, so hydrocodone and acetaminophen. It has shown phase 3 efficacy with really excellent safety profiles. If we look at the molecule itself, it has 4 contiguous stereocenters, I think one of which is quaternary. And so if you just kind of look at, the chemistry behind this molecule and just look at the beauty of it, I think also, carries its own weight. And then, you can also look at this story at a different angle, where, NaV1.8 was a seemingly less attractive pain target for many years. You're really taking a back seat to NaV1.7 as a target for pain, and then all of a sudden, NaV1.8 became really the hot area that companies start looking at. And then in this article, we have a series of articles looking at the NaV, the NaV as a target and the various compounds that have been produced in this space. But Vertex has been involved in a really long time in this space. And so, we cover, the whole 2-decade backstory of Vertex's, role in this space. Some other types of articles I'm super interested in and, really love producing are, the molecules of the month and the roundup articles. And so these are really short vignettes of the most current and cutting edge drug discovery science that's really ongoing right now. And in one article, you could read about a drug that was just approved. And in the next section, you could learn about a new inhibitor for a really hot new target. And so, you can kind of get a glimpse of the entire life cycle of drugs and, the drug discovery life cycle just in one article. And that's really what I find super interesting about some of these roundup articles that we do.
Yeah, no, that sounds definitely awesome. Yeah, I mean, in this opioid epidemic, like where people find, on playgrounds, even like opioids in San Francisco that children ingest, like something is really urgently needed. And yeah, the roundup articles are super exciting as well. Before we talk to Shema, I want to remind the audience that you can submit questions in the Q&A box. So at this, we're open for questions as well. I'll be starting to take some questions later on. So please feel free to populate the Q&A box already so that we have a choice of questions. Currently, I don't see any questions submitted. With that, I also want to talk to Shema. Hi, Shema. How's your day going so far?
Hey, Dennis. Happy Friday, everyone. My day is going great, actually. I really like that all of us are being here today and speaking about Drug Hunter and our favorite coverage at Drug Hunter. So thank you so much for you and the other team members for organizing this event.
Cool. Yeah, no, you're very welcome. And yeah, thanks for being a part of this great team. I always love working with you. Like all the articles I worked with you together, like you're always doing such an amazing job with, like writing things and providing things on time. Really amazing. I wonder, what is your favorite article that you've written so far in your career at Drug Hunter?
Dennis, this is a really tough question because I really love every single article that I write. I invest even like sometimes emotionally with my articles.
I bet. Yeah.
Yeah, I really love them. Sorry. But to pick one that was very recent as the mini pig, the rising role of mini pig in drug discovery. Sorry again. In this article, we explored several aspects of using mini pigs and preclinical research. So basically, mini pigs are a smaller version of the regular huge pigs that we all know, and they weigh almost 20 to 40 kilograms. What's really interesting about mini pigs is that they offer several advantages in preclinical research over the traditionally used non-rodent models, such as dogs or non-human primates. To name a few of these benefits, I would say, or I would mention the ethical considerations with using dogs and monkeys, and the cost, especially with the recent ban in 2020. So China banned the export of research monkeys. And according to a recent Nature publication, the cost of a monkey before the pandemic in 2020 was like 8,000, but now it could reach up to 55,000. Yeah, so the prices are just soaring high, and it's really important now to start looking for alternative whenever applicable. And mini pigs could be alternative in some scenarios. So in that article, we highlighted mini pigs as an excellent model for dermal drug studies due to physiological similarities between pigs and human skin. We also provided an overview of the use of mini pigs in a small molecule preclinical drug discovery. We discussed like several aspects and hepatic metabolism, oral bioavailability, CNS penetration, and toxicology. And as a bonus, we provided a guide on selecting non-rodent species with a focus on mini pigs in that article. And by the way, Dennis, through our letters to drug hunters, we received a request from our readers that they want to know more about rabbit PK, which is coming soon. So that's going to be really exciting.
Nice. Yeah, that's super exciting. Yeah, I know. I think also the mini pig got inspired by a reader as well. And I remember during my time at Novartis, my old advisor, my old boss, he was always talking about mini pig PK. Yeah, in Europe, everybody does mini pig PK. He worked at Boehringer Ingelheim, so it seems like this is a really so interesting topic to cover. Shema, how about your favorite recent molecule? Can you tell us maybe a little bit about, a molecule that you recently found interesting and some, transferable insights that the audience can take away from that?
Yeah, sure. And actually, I'm gonna like circle again on GLP-1 agonist. So my favorite molecule is orforglipron. Um, I like it because it is the most advanced GLP-1 agonist or the non-peptide GLP-1 agonist in a clinic. It's like first, it's not peptide. If we look back at Ozempic or Wegovy, or sorry, it's the same brands. It's semaglutide. If we look also at Saxenda, which was even used before Ozempic for weight loss, these are all injectable peptide GLP-1 agonists. So orforglipron is I would say it's a relatively small molecule because it has a molecular weight of 800. Also, it's oral. Eli Lilly has published positive clinical data last year in diabetes and weight loss, and it's currently under development in phase 3 in the same indications. What is really remarkable also about orforglipron, its physicochemical and pharmacokinetic properties. It has a bioavailability of almost 40% in rats and almost 25% in cynomolgus monkeys, despite the fact that it has a molecular weight of almost 800, over 800. It has a total polar surface area almost 140, and it has at least 8 ring systems and 15 hydrogen bond acceptors. So this is pretty remarkable, and it's just interesting to see that this is an oral bioavailable molecule.
Yeah, definitely super interesting. Yeah, you wouldn't expect that. Looking at this molecule, you definitely would not expect this to be orally bioavailable.
Yeah, I remember when I worked with Dennis on this, the first thing he was like, "Is that orally bioavailable?" I was like, "Yes, it is."
I almost didn't believe Shema. I had to have her check like 7 different times. Like, are you sure? How about in here? And how about here?
Yeah, and even the interesting thing is that it is without any absorption enhancers. For example, there is a peptide GLP-1 agonist, semaglutide also. They formulated it in oral formulation, which has they utilize the absorption enhancers. So it requires a special administration considerations, which is not supposed to exist with orforglipron.
Yeah, super interesting. Yeah.
Well,
I guess.
Yeah, that's awesome. Well, I guess we haven't done you, Dennis. What about you? What's your favorite, molecule, article?
Yeah, no, that's good question. No, really good question. I was like, I'm thinking about this. I'm gonna go back to the concept of covalency that Lou brought up a little bit earlier. So I really like Relay Therapeutics lirafugratinib, which is a selective FGR2 inhibitor. So I like that compound first because it was almost deemed impossible to find an FGFR selective inhibitor. I mean, of course, on the market there are pan FGFR inhibitors, but there are obviously dose limiting toxicities with those molecules in the clinic. So people were thinking that it's actually impossible to discover a FGFR2 selective inhibitor because the sequence homology in the active site of FGFR2 and FGFR1 is 100%. So they are 100% identical. But then the approach that Relay Therapeutics actually used was a molecular dynamics approach. So they leveraged a supercomputer called Anton. They worked together with D.E. Shaw on this and discovered that there was a P loop that contained a cysteine in that FGFR pocket, and that it would move faster in FGFR1 than it moves in FGFR2. So that makes FGFR2 more accessible by using a covalent warhead to the alkylation of FGFR2, which is super cool. The entire concept of kinetic selectivity I find super fascinating, and I think that's something that's also now been leveraged by multiple compounds in clinical trials, as well as even approved compounds like ritlecitinib from Pfizer. So this concept is just phenomenal. And interestingly, so when it also translates, the selectivity also translates into the clinic. So Relay Therapeutics is currently looking at this compound in cholangiocarcinoma. And typically, FGFR1 inhibition is associated with hyperphosphatemia, and FGFR4 inhibition is associated with diarrhea. And those side effects are currently not seen in the clinical trials, which is a sign that the compound may be FGFR2 selective. And then also the response rate, the overall response rate that was observed in a phase 1, phase 2 trial is over 80% in cholangiocarcinoma, which is remarkable, I think, given that the pan FGFR inhibitors have shown way lower response rates and were discontinued by patients due to those side effects. So I think that's a really fascinating story and definitely one of my favorite molecules that we recently covered. as for the favorite articles, I'll go with the patent highlights, actually. So I think that's something, that I haven't seen anywhere else covered than the way we cover it. So what we do, we like have a process of filtering down all the patents that, are published every month, and then have a really rigid editorial process in picking, maybe the top 10 patents that we then provide structures and biochemical data, DMPK data on. And in the end, we have like a searchable table that you can jump through and monitor specific targets, specific companies, or like disease areas you're interested in. So that's something that I really love, because it allows you to stay on top of the literature. So whatever is published in patents today will probably not go in the primary literature until maybe like in 5 to 10 years. So you can really stay ahead of the curve by following those patent highlights. Really, really love those. Cool.
Awesome
I
choices.
think at this point we're open for any questions. I haven't seen any questions come through. Maybe I'll ask Lou a question actually in the meantime. So Lou, can you tell us a little bit more about what is going on in your department right now? Is there, anything that you want to maybe share with us?
Yeah, so if there's anybody out there who would love to join this exciting content team, we are looking for another scientific editor or senior scientific editor. So if you have a passion for researching and reading and learning and writing and really sharing important drug discovery insights with the rest of the community, and want to try to help accelerate the development of medicines in this way, please apply. We just opened this position up, I think, last week. We've got a lot of interest in this position, but really we'd love to hear from you and get a sense of if you'd be a good fit with the rest of the team here, joining me and Shema and Rory and Matt as part of the content team.
Awesome. Yeah. I can totally echo that. It's actually, I love working with everybody on the team. Like it's like we have such a nice culture here, I think, at Drug Hunter that's totally unique. I mean, I felt working at Novartis and Emeryville had a special culture as well. But yeah, the culture we have here at Drug Hunter is like outstanding in my opinion. We do have a question from the audience now that I'll just shoot out to the panel. I probably am not pronouncing this right. Muxia is asking, in your experience and analyses, what are some of the most promising new technologies or approaches being used in drug discovery today? Does anybody want to come out with an opinion? If not, I can jump in.
I feel like this is a great roundtable question. Maybe everybody should just say one thing that they're really excited about. How does that sound?
Perfect.
Yeah. You want to go first and then pick the next person?
Yeah, no, I'll go first. So I'm really excited about targeted protein degradation. I think targeted protein degradation is currently still in its infancy. We don't have an approved drug on it yet. We may be really, really close. I think just the potential of that technology that it offers, that it so much differentiates from just inhibiting a specific protein, I think it's just super fascinating, and I could talk on about this forever, but of course we want to give everybody the chance to name a technology. So I'll pass it on to Matt Hesse.
Oh, thank you. I mean, I think the obvious one, which is kind of the elephant in the room, is the rise of the use of AI, right? And I think there is probably a lot of hype around it, but we're really starting to see some concrete steps towards it being used. And like any tool, it's appropriate to use. You have to work out the right situation to use it in. But we just had an article on Schrodinger's first in-house asset using their. I mean, Schrodinger's been around for 30 years in computational chemistry. But they really showed how they can leverage that in order to get to a clinical candidate. And I think they triaged something like 8 billion compounds, which were generated from their kind of computational workflow and then use the multi-parameter optimization to really drill down on these are the guys that we want to put synthetic effort into synthesizing. What eventually became their clinical candidates got a pretty difficult quaternary stereocenter. And so the team, they investigated something like 11 different synthetic routes to get to that, which, that's a lot of faith in this kind of computational prediction. But I think that speaks to kind of how powerful it's become that you're willing to invest that amount of synthetic effort into because you really believe that it's going to give you a superior clinical candidate.
Want to pass it on to the next person, Matt?
Sure. Let's. I think Lou looks like he's primed.
I have so many, but I'm going to select one. I'm going to select the advances in chemical synthesis that have been happening and are continuing to happen. I've just been astounded that since I left graduate school in 2002, the landscape of chemical synthesis has completely changed. To be perfectly honest, being somebody who's more consumed with drug design and, translational sort of sciences mindsets as you're doing lead optimization programs as a more senior level drug hunter, a lot of the chemical synthesis advancements I'm sort of looking at from the outside, but I'm still just really blown away with them. And one in particular that I think is incredibly exciting is this area of synthetic chemistry called skeletal editing. This is something that really just cropped up in very recent years. I mean, some of the transformations that are occurring out there were defined by people like David Rees and others as dream reactions as recently as 2018 in Nature Chemistry. And that dream has already come alive with CH group to nitrogen atom exchange and aromatic ring systems through the work of people like Mark Levin. I gave a talk on this a few years ago, the necessary nitrogen atom, and I talked about this dream reaction and how great it would be to be able to do this type of skeletal editing. And 3 years ago, I thought this might happen in 20 years. Well, it happened in less than 3. So I am blown away by the power of synthetic chemistry and its ability to help us make higher quality, more novel, more complex, and a greater variety of drug molecules than we've ever been able to make before.
Yeah, that's awesome. Great pick. Yeah, it's pretty crazy. I noticed also, Lou, you did your PhD. It looks like you co-authored a paper with Dave McMillan.
So,
right?
Yeah, I had the privilege of taking over Dave's project. So those were big shoes to fill in graduate school. Dave thanked me at one point for making him look good. So I think I did it.
That's awesome. Yeah, speaking of synthetic innovation, I mean, just the stuff coming out of all the groups like that are absolutely incredible. You want to pick the next person, Lou?
Rory.
All right, cool. You're very bashful, Lou. I mean, I feel like you could take a lot of credit for the skeletal editing field and all the science papers and nature papers that have come out of since your necessary nitrogen perspective. So, but I really love your answers. Synthetic innovation is really awesome. In terms of other technology, I have to say perhaps Dell. Just as a plug, we have a Dell article coming out here probably in the next week and a half or so. And so up to this point, there's been 4 clinical compounds that have been identified using Dell or enabled by Dell, and I think there's 2 others that haven't been publicly disclosed yet. So it's quite impressive to see really the impact that Dell is having both on the clinic, but also as a way to really start reaching novel chemical space and novel chemical matter. This area of technology is just quite impressive to me, and I really look forward to seeing what else it produces. Do we see Shema yet, or Shema, I think you're last then.
Yeah, thank you, Rory. So my favorite would be the chemoproteomics. Actually, we covered this twice recently, and I really was amazed by this technology. It utilizes the compound libraries of diverse scaffolds and covalent handles, and it's then used to scan an entire proteome to identify novel pockets. And I really liked how David Weniston, he described this in the ACS spring meeting as the Tinder of drug discovery versus arranged marriage, which traditionally the scientists have been doing. So it's just like they screen those like huge amount of compounds against the whole proteome, and then they measure the binding affinities and just to detect which compound binds to the target. So I think this is really important because it allowed us to discover new binding sites. Also, as we've been like mentioning, that it allows drugging the undruggable. So that is really an interesting platform, which lead to the discovery of keep-on inhibitors and Warren helicase inhibitors. So I think this is a pretty promising and exciting technology. Yeah.
Thank you, everybody. Let's hear Dennis's favorite.
Oh, my favorite. Well, you guys, it's funny because as you were going through and picking each one, you were each taking the one I was going to say. So now, but I would say, one other one that we haven't talked about yet is mRNA display screening. So mRNA, and specifically for, macrocyclic peptides. So mRNA display obviously has been used to help discover biologics, antibodies, things like that, to come up with this chemical matter that can target really challenging protein surfaces. Like you can target protein-protein interactions, find starting points for all kinds of crazy things, KRAS. And I think there was, for a long time, even though this technology had been available to generate macrocyclic peptides, people kind of, dismissed it as, oh, well, you're only going to be able to find tool compounds with this. You're never going to get these things in cells because they're super polar. You're never going to make these things orally bioavailable. And now, all of a sudden, we've got several oral drugs that have been derived from mRNA display screening or our macrocyclic peptides in that space. So, I think this has opened up the ability to target surfaces that were previously thought to be untargetable. We talked about PCSK9, we talked about KRAS. Now there are molecules targeting IL-23R that aren't necessarily derived from mRNA display screening, but are in that chemical universe. So I'd say that's an area that I find super exciting.
Definitely, yeah. And we've seen approvals from that area as well now, which is super, super exciting too.
That's right. First approved drug from mRNA display screening last year. So great article on Drug Hunter about that.
Yeah. So yeah, speaking about articles on Drug Hunter, maybe before we wrap it up, let me show everybody, since now we talked a lot about our favorite molecules, I want to show everybody how one of these articles looks like on Drug Hunter. So we can just search for, so the cool thing is we have like a full text searchable content here. So we can basically just search for Dennis's favorite molecule. And as you can see, we have an article here and we have a molecule case study here. And I just want to show everybody the depth of the case studies that we're running. So basically, you can appreciate how much we cover. So Dennis already talked a little bit about the concept of mRNA display screening. You can have a very nice visual overview of that here. We cover really the compounds, the starting points, like from hit finding, we start to the lead optimization, like here, really some interesting co-crystal structures that were absolutely necessary to advance this program into a clinical stage. And you can just see how mind-boggling these structures look like. It's just absolutely insane that Merck was able to move this program into the clinic, and not only that, see antibody-like efficacy in the clinic with these types of molecules and make them orally bioavailable. So just absolutely mind-blowing program to me. So yeah, you can see the entire depth of that program here. We also highlight synthetic highlights in the end to lose point. There's always new innovations in synthesis, and we try to cover this in our articles as well. with that, I think I'm not seeing any more audience questions. So maybe it's a good time to wrap up here.
Well, there is
We're also coming up on the hour.
I do see an anonymous question, Dennis. I'm not sure if you can see it from an anonymous attendee. So the question is, does Drug Hunter offer individual subscriptions for drug hunters who may not be affiliated with an institution? While Drug Hunter was developed to help active drug hunters, it could also be a very valuable resource for those who do not actively work as med chemists in the industry. So people in academia recently laid off, etc. So yeah, a quick answer there is Drug Hunter is primarily intended for people working in industry. So it's usually companies or institutions like the NIH or places like that that will subscribe on behalf of their users. We do have a program for anybody who's been recently laid off. So if you used to work in biotech or pharma, but were laid off and are currently seeking a job in biotech or pharma, you can apply for one of these transition programs that we have. So stay tuned. You can check on the website for that shortly. At the moment, we don't have personal subscriptions. I know the team is working on trying to pilot some kind of expanded access program later in the year, so stay tuned for that. But at the moment, if you are interested, we are primarily serving groups affiliated to institutions.
Awesome. Yeah, thanks for answering that. Sorry, I missed this, Dennis.
No worries. It might not show up because it's anonymous. I just saw it on my screen.
Oh, there's actually one more question coming in that I see right now. So maybe we can take that with a short answer and then wrap it up. What is the future of covalent inhibitors and multi-target ligand designs? Is the question. Maybe I can start talking about this a little bit. It's of course, a big question. I think we're seeing a comeback of covalency, like a lot of compounds that like covalency was initially, of course, thought about with like really high safety hurdles, maybe something that can only be applied in oncology indications. But we're seeing even covalency being used as a concept outside of oncology indications, with the redlicetinib as a prominent example in a hair loss condition, chronic autoimmune hair loss condition called alopecia areata. So there's definitely something there, and I think the industry is witnessing kind of a commercial comeback, a more of an acceptance for covalent inhibitors, and really excited to see what the future holds. In terms of multi-target ligands, not sure there. I mean, if you consider, for example, pan-RAS inhibitors as multi-target because they target multiple different forms of RAS, there's certainly some said there, and as Matt highlighted in this, particularly the RevMed molecule could be something to watch out there. Anybody wants to add something before we wrap up, or?
I think that's fantastic. We're just at the hour, so thank you everybody for joining us. For those of us who dialed in live, it's wonderful to see you. And for the team, thank you so much for participating. I think this was a really fun discussion, very thought-provoking. I learned a lot from each of you, and looking forward to all these unpublished articles that you mentioned. Can't wait to read them myself. Oh,
awesome!
Raised hand from Yajun. Is there something in the chat there?
Oh, no, not that I see. Oh,
maybe he's just waving.
Yeah,
maybe just waving us. Okay.
Hi,
yeah, I also want to thank everybody on the editorial team. Thank you guys for a really engaging discussion. Also, thanks to the behind the scene workers at Drug Hunters. Those are the freelancers, our marketing team, our operations team, our sales team. You saw only a fraction of the Drug Hunter team here on the call today, and really everybody pulled together to make this event happen. So really appreciate that. And then I want to put a little plug in. Next week, we actually have a flash talk on generative AI. Matt highlighted as one of his favorite technologies, AI. So if you are really curious about this, dial in next Thursday. with flash talk from Ashwini Gorare from Merck, Millipore Sigma. She will basically give a talk and a basically overview, a guide for medicinal chemists, how to use generative AI. And then also stay tuned for more flash talks on Fridays. We're planning to do more of these where we would share strategies, how to overcome certain problems that you may be having on your drug discovery campaigns. And you can also learn how the CEO himself uses Drug Hunter to stay on top of the literature. And with that, I want to say bye to everybody and have a happy Friday. And we look forward to seeing you next time.